Hi, my name is Alicia. I work on mitochondrial dysfunction as it is a biomarker for MED13L syndrome. I just finished up my PhD in Dr. Randy Strick's lab. So he spoke earlier about cyclin C. Hopefully we remember a little bit of the molecular cell biology from before, because I'm going to do a little bit of a deeper dive into it now, and specifically with mitochondria. Okay. So another player of the mediator kinase module, let me pull up a pointer, okay, is cyclin C, which Randy mentioned earlier. It's part of the same complex as MED13L. This complex, which has been introduced a few times today, is the mediator kinase module, or MKM, that consists of an even stoichiometric ratio of the following components. So cyclin C, MED12 or MED12L, MED13 or MED13L, and CDK8 or CDK19. Canonically, our work and others have shown that the MKM represses and activates approximately 3,000 mammalian genes. Interestingly, there is about an even split between those that it represses and those that it activates. We have also shown that cyclin C, this protein here, has a secondary role, or it has a dual role in the cell, in both controlling mitochondrial dynamics as well as the canonical role in transcription. So as I mentioned, cyclin C has a dual role in the cell. We found that in response to oxidative stress, or more importantly in this case, in the presence of a mutation in another MKM component, MED13L, this led to the nuclear release of cyclin C, meaning that cyclin C is now mislocalized to the cytosol of the cell. Cyclin C, we showed, directly interacted with a mitochondrial fission partner, factor DRP1, and our hypothesis became that if we had continual mitochondrial fragmentation and continual cyclin C release, this would ultimately lead to a decrease in mitochondrial fusion, a decrease in mitochondrial membrane potential, which is needed for ATP production and energetics in the cell, and a decrease in mitochondrial DNA copy number. And potentially, this could be a molecular mechanism for what we are seeing with neurodevelopmental syndromes, most importantly MED13L syndrome. So this is indeed exactly what we showed, mitochondrial fragmentation, in our first publication when assessing patient cells from a collaborator in the Netherlands, Dr. Van Hasselt. We obtained a cell line from a skin biopsy, much like many of you have participated in recently, more skin biopsies. We were able to show that there was an overall reduction in mitochondrial dysfunction that corresponded with organelle fragmentation. A huge caveat to this study was the fact that we only had one patient sample, an individual with a thymidine duplication in exon 20, leading to a frameshift mutation and ultimately truncation mutation after exon 20. So the goal then became to further characterize additional MED13L patient samples, all with varying mutations and severity of phenotypes. So this is in collaboration with Dr. Bain, who spoke earlier at Columbia. Some of you may have taken the trip up to Columbia to do the skin biopsies. With the help of the MED13L Foundation to round up a bunch of people, we were able to collect a variety of different samples with different mutations and locations along the MED13L coding region. Additionally, at the time of this study, Dr. Bain performed behavioral analyses on 8 of the 12 individuals, looking at gross motor skills, fine motor, and manual dexterity and communication skills. Some preliminary analysis that I did with this was looking at different types of mutations in MED13L, and this revealed that tasks related to movement were slightly more difficult for individuals with deletion mutations. However, those with frameshift mutations had more difficulty with communication. It's currently in the works. A lot of people have talked about it today to compile parent-reported behavioral analyses and hopefully correlate that in the future with genotypes. So in terms of the 12 additional patient samples, I mentioned that there are a variety of mutations included, as well as location along the MED13L coding region. So I will add up all the mutations here. You might recognize your child as one of these mutations up on the screen. There are also several regions represented along the MED13L coding region, including the N-terminal, C-terminal. There are several intrinsically disordered regions, or IDRs, throughout the protein, as well as the mid-med-PWE region, which is an RNA binding motif. And as you can see, we have several deletions. A couple of these are ones that Matt was actually just talking about, so the P8L, 866L, exon 3 through 25, and a variety of locations. So further, if we break it down by group location of mutation, we can break it down into different regions. So we have N-terminal located mutations, C-terminal, mutations that are in the mid-med-PWE region, and then those are what I'm deeming the middle region. I also made sure to include that if it overlapped with a specific IDR or leucine zipper, for example, to break those into their own categories. So as our original studies indicated that a mutation in MED13L led to cyclin C nuclear release, mitochondrial fragmentation, and dysfunction, it was of interest to determine if this molecular phenotype was conserved across all other variants. As a control, I have human dermal fibroblasts. This is indirect immunofluorescence, looking at red are the mitochondria in a cell. Green is cyclin C, and you can see that cyclin C is entirely in the nucleus in this control cell line. And I will add up these pictures of, these are 4 representations of the 12 different cell lines. I have one missense mutation, a frame shift, nonsense, and a deletion. But more importantly, you can see that all 12 patient cell lines had this exact same cyclin C release and varying degrees of mitochondrial fragmentation. So cyclin C is, again, in the green, and areas of yellow indicate an overlapping of cyclin C and the mitochondria. So again, what we had previously shown with our 1 patient sample, we were able to recapitulate that with all 12 patient samples. I next assessed mitochondrial fragmentation. This was more of a complex phenotype, so not all cell lines had the same amount of fragmentation. But what I did was I grouped it together based on type of mutation, again, trying to make that relationship between genotype and phenotype. All had significantly more mitochondrial fragmentation compared to the control. However, there was maybe slightly less in the nonsense, and nonsense and deletion variants were statistically different from each other. Lastly, I wanted to understand if these mutations in certain locations led to more cyclin C release and therefore more mitochondrial fragmentation. So this is grouped based off of N-terminal, C-terminal, all the different regions that I was showing on the protein MED13L. And you can see that mutations that are earlier on in the coding region had more mitochondrial fragmentation. So it's possible that these are protein truncating variants that do not have the ability to tether cyclin C in the nucleus anymore. It's well known that mitochondrial fragmentation can lead to an increase in both local and cellular ROS, or reactive oxygen species, as we heard from Randy before. We have shown this previously in the 1 MED13L patient sample, so again, the whole theme of trying to recapitulate this in all 12 patient samples. I first used DHE, which is a stain to look at cytosolic ROS, and this is flow cytometry, with a few of the cell lines, and you can see that a couple of them are more pronounced than others in terms of an increase in overall cytosolic ROS. The same experiment was repeated with mitosocs to look at mitochondrial ROS specifically, since we are dealing with mitochondrial fragmentation. And again, the same pattern was seen here with 2 of these variants maybe having a little bit more mitochondrial ROS than other variants. This data right now is being expanded to include all 12 of the patient's cell lines. So this is just a representation here of ones that have gone through all of the biological and technical replicates to make this statistically significant. So it was also of note that compared to control cells, and if I show a picture, it'll be a little bit easier to understand, that the patient's cell lines appeared to have less mitochondria overall. It's nice to look at a picture, but again, we need to have some sort of quantification for this. So I actually used one of the software packages with our fluorescent microscope to help quantify mitochondrial volume. So it isolates the individual mitochondrial particles and can calculate the number or the mitochondrial volume as a whole for a variety of cells, average it together. So you can see that there were significant reductions in, I have 3 cell lines here that had less mitochondrial volume compared to control. So what we're seeing quantified here is exactly what I was first noticing when I was taking the images of the cells. As I had identified an increase in overall mitochondrial fragmentation in cells harboring MED13L mutations, it was important to determine if the mitochondrial networks themselves were still functional. So this, I used seahorse metabolic flux analysis, which can test for different ATP rates in the cell from different sources. So whether that is through glycolytic ATP or mitochondrial ATP through the electron transport chain, we can use a variety of different inhibitors. So this starts with oligomycin, and then a combination hit of rotenone and antimycin A are applied to these drugs, and real-time measurements of mitochondrial function can take place. So cells are living in a dish, and I can measure mitochondrial function as these cells are living. We can also break it down by these 2 different types and determine that maybe one type, for example, has more energy overall, but if you look at it, these cells are more glycolytic, which is not necessarily indicative of a healthy mitochondrial network and a healthy cell. So what I did was I did this for all 12 cell lines. It's a lot to look at on one graph, so again, I broke it down into the different types of mutations, and all of them, except missense mutations, had a significant decrease in mitochondrial activity. This is looking at oxygen consumption rate, so this is mitochondrial ATP specifically. And then lastly, I grouped this based on location, and unsurprisingly, again, N-terminal mutations had less mitochondrial output overall. So now we're starting to kind of put together different pieces here of what genotypes correlate with what phenotypes we see at the cellular level, and then being able to apply that to data we have from other researchers who maybe focus more on more behavioral studies and being able to tie all of it together. I also wanted to understand the functionality of the mitochondria a little bit further, so I did the metabolic analysis, but now I'm looking at mitochondrial DNA. Randy hinted earlier that mitochondria contain their own mitochondrial genome in the form of this circular small genome that encodes 13 proteins, and it can actually be an indicator for overall mitochondrial health. So again, I love the pretty pictures of the microscopy. You can see that in the control cell line, red again are your mitochondria, green is a stain for double-stranded DNA, which can actually show your mitochondrial DNA. There is a drastic difference between the patient cell line that was analyzed here and the control. Not only were there less mitochondrial DNA copies overall, but we also had stretches of mitochondria that did not have mitochondrial DNA. This does not necessarily mean that there are mutations in the mitochondrial DNA itself, which makes it a very therapeutically viable option. I also wanted to quantify this because pictures are nice, but quantification is even better. There is a very, very strong phenotype here when looking at mitochondrial DNA copy number. This is an RT-qPCR-based method that gives relative amounts of mitochondrial DNA compared to a single genomic locus, typically something like pyruvate kinase that only has one locus throughout the genome. And you can see that this is very significantly different for all of these. And lastly, as the cells were maintained in culture, I started to notice that they were growing a little bit slower. And one of the things that I've started to test, this is all preliminary data, but is something that is ongoing right now, was looking at premature senescence, which is something that is a hot topic in aging studies, so how quickly these cells are reaching their limit. An overview of this, just for the sake of time, is looking at the number of cells that are still actively dividing, and I noticed that early on, a few of these cell lines were starting to decline in terms of their division. So through this work, we were able to show that mutations in MED13L have an impact both at the transcriptional level and organelle level at the mitochondria, and that these transcriptional defects, as well as the aberrant cyclin C nuclear release, are not just limited to one mutation type, but are common with all mutation types, which is something that we will be talking about in another presentation when we look at repurposing drugs to help combat mitochondrial dysfunction. So if you hang on a little bit longer, I'll have that talk. And another pretty picture. But thank you for the invite to come talk today. I'm always willing to talk to people about science. I think it's super important, science communication, to bring what might be scary in a white lab coat in a lab to the general public. So feel free to ask me any questions. Nothing is a stupid question.
Good job. Okay, we have time for a couple of questions for our speakers. Anybody have any questions for our last speakers?
This is really a question for all three speakers. So, with the work that you're doing, how close do you think we are in mouse models or in mitochondria cell models to having assays where we can test therapeutics and have a phenotype or a biomarker that we can really use to test whether or not medicines are working in mice or in cell models?
So in terms of the mitochondrial work, we're there already. We have a biomarker that is cyclin C nuclear release, mitochondrial fragmentation. So any sort of therapeutics that are applied at the cellular level, we would be able to see a potential rescuing of mitochondrial fragmentation and overall function. So that's what I'll talk about, the drugs that I've chosen and am working on currently, applying those to the cell model. So that is more so just a biomarker for mitochondrial dysfunction. In terms of the animal models, I don't know if Matt...
Yeah, we'll let Matt finish that one.
And so where we are with the mouse models, I would say we're right at that point where that conversation needs to start happening. So we have the animals, they're viable. As I mentioned, they have genetics that are very conducive for genetic therapies. The only downside, as I was kind of showing, is it's kind of specific to either one sex or one allele type. But that being said, the way they're approaching genetic therapies like base editing, like replacements, like ASOs with the FDA right now, is they understand that a mouse is not a human being. They're not always going to be one-to-one. And in the case of neurological disorders, mice are very different from human beings as far as the way their brains are developed. So oftentimes we see mice are either more severe or less severe than the human disease. And the FDA is starting to really understand that. So what we can do is leverage phenotypes that we see in cell models. Say we have biomarkers here that are directly related to the gene's function. And then we can use the mouse models to develop and screen, and then to show efficacy, or not efficacy, but safety. So there's been several gene therapies that have made it to clinic recently. One of them went from diagnosis to clinic in 9 months. And it's because they were able to show the mouse model they had was not going to be anything that can give us any phenotypic readouts or efficacy readouts, but they had a lot of cellular work that was already in the process of being made when the diagnosis happened. And they were able to say, look, we can fix it in the cells. The way it works in the cells is very well known. We have good pathway, good mechanism there. So all we needed to do was show that in the mouse, it was safe, that the mouse didn't grow a second head. And then they said, that's all we can really do. That's all the tools that are available, and there is no other solution for these kids. So they were able to argue compassionate use, which is a doctrine the FDA picks up when there is no way forward, that the prognosis and the outlook for the patient is just nothing. It's like there's no quality of life that's going to be improved in any way, shape, or form. So that's kind of where we're at right now, is we have the animals, we have some cell line work that can be used and leveraged in that case. So I think it's in a good place to start moving that conversation forward.
For you, all set. Anybody else? One more question?
Just a small comment and question to follow up on both of those. So I think the mouse work is really important to compare and contrast your findings, you know, of what's happening at Jackson, what's happening at Iowa, and looking at what is reproducible even across institutions. Mouse studies are tremendously challenging to reproduce, and mice are just annoying. Some mice you put into studies and they just won't even follow along, and you got to exclude mice, and so they have to be super well powered. But it helps to have this, and I got a sense that you guys were both kind of hinting at this anxiety phenotype, which was very encouraging, and even the sex specific and strain specific backgrounds, all this really contributes. So I think it's fine to have a sex specific effect so long as it's reproducible and you guys are finding the same stuff. That's super encouraging. I'd be curious just to know on that, are you going to focus and do just better anxiety assays, like a elevated plus or a zero maze, an elevated zero?
I did the elevated, or at least I'll let Matt speak about his specific mice. But for the, I did an elevated zero maze, and I didn't show the data because I didn't observe any statistical differences between the hets and the wild types in either case. It looked like there could be a trend in like a decrease in the total distance traveled, but again, it wasn't statistically significant. So the open field test was where I saw the most significant data results.
Yeah, and the follow-up on that, we looked at a couple of different approaches to anxiety, and while we do see there's a hint of it there, it's not particularly overwhelming. And to give folks here an idea, when we develop these mouse models for therapeutic testing, as a person who runs a lot of those type of therapeutic testings, I'm looking for phenotypes that are very differential, like the distance between the wild type and the mutants. You're seeing at least 2 asterisks, preferably 3, because what that means is that's going to be really well powered. Running behavioral assays in mice, especially for therapeutic developments, is kind of costly. You don't want to have to run 20 or 30 animals per test arm in order to see a difference. So you want something where you can run, let's say, 5 to 10 animals and see, let's say, a 20 or 30% improvement. Because the other thing here is we don't expect to see 100% rescue with any drug or any therapy. So you have to be able to statistically see that you're moving that needle back towards healthy without having to run a zillion animals to resolve the statistics on that. So for my mice right now, I think probably the strongest phenotypes I have that would be conducive to that and are replicable is the wire hang result for the 861L, as well as the cholesterol that we're seeing there. But that drags the question back towards, what does that mean then towards the patients? Because that's the first thing the FDA would say is, what does the cholesterol and a wire hang phenotype have to do with MED13L syndrome? So we could argue it on the wire hang from being a strength, so muscle tone, and that could be also connected back to the mitochondrial stuff as well. So there's a little bit more work that could be done there. As far as other more anxiety-based or behavioral-based things, one of the things that I am discussing with my team on running is something called an acoustic startle assay, basically how much an animal flinches when they hear a loud noise, as well as a follow-up to that, which is a kind of a preconditioning where you give them a loud noise and then you give them a slightly lower noise and see if they respond to the loud noise as extremely. So those are things that we're going to look into there. But like I said, you don't always have to have the strongest mouse phenotypes if you do have good cellular work that backs up mechanism as well. So between the two of those, you can combine them together and put together a pretty attractive package for the FDA to review and say, we understand the limitations of what the models can do. We understand where things are at. And because of a lot of the work that's already been done by other groups in the path for genetic therapies, they understand where the real crux parts are. And usually that revolves around safety. At the end of the day, if there's nothing else that can be done, they just want to know, if we go to clinic with this and we put it into a kid, the worst that can happen is it does nothing. That's not a terrible output if there's nothing else available. But we have a little bit more work to do just to make sure that we've tried everything that we can do and see where we get from there.